Droplet libraries are collections of tiny discrete droplets, often on the nanoliter to picoliter scale, that serve as miniaturized reaction vessels for high-throughput research. By compartmentalizing reactions into individual droplets, scientists can conduct thousands of experiments in parallel, significantly accelerating discovery in fields ranging from chemistry and biology to materials science. How are these libraries created? Typically, microfluidic devices are used to generate uniform droplets containing reagents, cells, or nanoparticles. For instance, by controlling the flow of two immiscible fluids, such as oil and water, in a microchannel, one can form droplets with precise volumes. These droplets can be designed to contain different chemical compositions or varying concentrations of reactants, forming a library that allows researchers to screen a wide range of conditions simultaneously. This approach is particularly valuable when exploring reaction kinetics, optimizing synthesis conditions, or conducting combinatorial chemistry. One advantage of droplet libraries is that they reduce reagent consumption and waste compared to traditional bulk experiments. Each droplet acts as an independent microreactor, and because of their small volume, reactions occur quickly and can be observed in real time using optical or spectroscopic methods. For example, in drug discovery, a droplet library might be used to test the effects of hundreds of different drug formulations on a target cell type. What makes this approach so powerful, the ability to rapidly test many conditions in parallel, not only speeds up research, but also uncovers subtle trends that might be missed in larger-scale experiments. Droplet libraries also enable precise control over experimental variables. By altering the size, composition, or temperature of individual droplets, researchers can systematically investigate how different factors influence a reaction. This high level of control is critical when dealing with complex systems where multiple parameters interact. In environmental science, for instance, droplet libraries can simulate micro-scale chemical reactions that occur in natural waters, providing insights into pollutant degradation or nutrient cycling. Advanced detection techniques, such as fluorescence microscopy or mass spectrometry, are often integrated with droplet-based systems to monitor reactions in real time. These techniques allow scientists to track the progress of each reaction within the droplets, enabling rapid data collection and analysis. How does real-time monitoring improve the research process? It allows for immediate feedback, which can be used to adjust experimental conditions on the fly or to quickly identify promising reaction pathways. Droplet libraries offer a scalable, efficient, and highly controlled method for conducting a wide array of experiments simultaneously. They are created using microfluidic techniques to generate uniform droplets, each acting as a microreactor. By leveraging these libraries, researchers can dramatically accelerate screening processes in drug discovery, material synthesis, and many other fields. How can droplet libraries transform research? They open the door to high-throughput experimentation with minimal resource usage, allowing for rapid optimization and discovery across a multitude of scientific disciplines. Droplet libraries consist of millions of microdroplets, each acting as an independent microreactor containing different chemical compositions, biological samples, or nanoparticles. These droplets are generated using microfluidic devices where fluids are precisely manipulated under laminar flow conditions. The advantage of droplet-based systems is their ability to miniaturize experiments, allowing for massive parallelization and high-throughput screening. Each droplet is isolated, preventing cross-contamination, making them ideal for controlled biochemical reactions. In drug discovery, droplet libraries allow thousands of compounds to be tested simultaneously in a single microfluidic chip. It's like a library of test tubes shrunk down to microscopic droplets, each carrying a different experiment. Droplets are generated in microfluidic channels using oil-water emulsions, where aqueous solutions form tiny suspended droplets in an immiscible oil phase. Precise droplet formation is controlled by flow rates, surfactants, and channel geometry, enabling uniform droplet size and reproducibility. Advanced methods like TJ junction, flow focusing, and co-flow geometries are used to tailor droplet properties. These controlled microenvironments allow scientists to perform experiments on a nanoscale with minimal reagent consumption. Biomedical researchers use microfluidic droplet generators to encapsulate individual cells in tiny droplets for single-cell analysis. It's like a vending machine that dispenses identical-sized water beads each containing a unique ingredient. Encapsulation in droplets creates tiny protective environments, shielding chemical or biological samples from contamination or degradation. Surfactants in the oil phase stabilize the droplets, preventing coalescence or evaporation. This technique is widely used in enzyme assays, chemical reactions, and bacterial culturing, where each droplet serves as an isolated laboratory. Droplets also enable controlled exposure to stimuli, allowing precise reaction monitoring over time. Researchers encapsulate single bacteria in droplets to study antibiotic resistance without cross-contamination. It's like storing tiny bubbles in a soap film, each holding a different ingredient safely inside. To keep track of individual droplets, researchers use barcode molecules, fluorescent tags, or digital droplet PCR encoding increments. For each droplet can be assigned a unique identifier, allowing real-time monitoring of its contents and reaction progress. Modern microfluidic platforms integrate automated tracking systems, ensuring accurate data collection and analysis. This technique is essential in genomics, protein engineering, and high-throughput screening, where thousands of reactions occur simultaneously. Scientists use DNA barcoding in droplets to track genetic mutations in CRISPR experiments. It's like placing a unique QR code on every tiny water droplet to scan its identity later. Droplets can be merged and mixed on demand using electrical, optical, or hydrodynamic forces, allowing researchers to initiate reactions at precise times. Controlled mixing ensures efficient diffusion of reactants, enabling chemical synthesis, polymerization, or DNA amplification inside individual droplets. Advanced droplet on-demand systems use dielectrophoresis, DEP, or optofluidic techniques to fuse selected droplets in microfluidic networks. This method is crucial for rapid diagnostics, point-of-care testing, and personalized medicine applications. Medical labs use droplet merging techniques to mix patient blood samples with diagnostic reagents for rapid disease detection. It's like clinking two tiny test tubes together to mix their contents instantly. Advanced microfluidic systems integrate fluorescence-activated droplet sorting FADs and machine learning algorithms to classify and analyze droplets in real time. Droplets containing desired properties, such as high enzyme activity or successful drug interactions, are sorted for further study. Automated AI-driven platforms can process millions of droplets per hour, enabling large-scale screening in biotechnology and materials science. The combination of high-throughput screening and AI-driven analysis is revolutionizing the efficiency of scientific research. Biotech companies use AI-powered droplet sorters to identify high-yield microbial strains for biofuel production. It's like a conveyor belt scanning and selecting the best apples based on ripeness and color. Droplet libraries enable the rapid testing of thousands of drug candidates, reducing time and cost in pharmaceutical development. By encapsulating patient-specific cells in droplets, researchers can analyze how individual patients respond to different treatments. This personalized medicine approach ensures tailored drug formulations, improving treatment outcomes while minimizing side effects. Droplet-based drug screening accelerates antibiotic discovery, cancer therapy development, and immunotherapy research. Cancer researchers use droplet-based assays to test which chemotherapy drugs work best for a patient's tumor cells. It's like having a mini lab for each patient, testing multiple medicines at once to find the best match. Beyond medicine, droplet libraries are transforming environmental monitoring, material science, and industrial chemistry. In environmental research, droplets are used for detecting pollutants, heavy metals, and bacteria in water samples. Industrial applications include self-healing materials, catalysts for chemical production, and nanoscale coatings. The ability to conduct rapid, small-scale experiments makes droplet libraries a cost-effective tool in diverse scientific fields. Water quality monitoring systems use microfluidic droplet libraries to detect contamination in drinking water. It's like miniaturizing an entire chemical plant into thousands of droplets, each performing a different test. The next frontier in droplet libraries involves AI-driven analysis, quantum-enhanced sensing, and bioprinting technologies. Future applications include organ-on-a-chip models, where human tissues are grown in droplets to test personalized treatments, researchers are exploring quantum sensing for extreme precision in biochemical reactions, revolutionizing detection limits. These advancements will push droplet-based research beyond its current limits, making experimentation faster, cheaper, and more precise. Scientists are developing 3D bioprinting systems using droplet libraries to grow artificial tissues for regenerative medicine. It's like turning each droplet into a programmable lab capable of running thousands of automated experiments.